happy to present today Professor Bjorn Bauer. Dr. Bauer is now working at the University of Kentucky College of Pharmacy, Department of Pharmaceutical Science in US. Dr. Bauer at present time is a Patrick P. DeLuca Endowed Professor at the Department of Pharmaceutical Science at College of Pharmacy, University of Kentucky. He has been faculty of different institutions, especially from University of Kentucky. About honors and awards, I have to say that he has a lot. He has a lot. It's difficult to present all the awards that Dr. Bauer has, but I want to say that he has the University Research Professor title. He is the top 200 world most cited researchers. Then he has been received a lot of citations. And now he is Roche Initiation Speaker Award. He has a lot of publications, and his PhD dissertation is very interesting because it was focused to in vitro cell culture models of the blood-brain barrier to study permeation of P-glycoprotein interaction of drugs. And this is a very important topic because can be focused to different drug-resistant conditions like epilepsy. At present time, his age index is 42, and the total citations he has received is more than 10,000. Then you can see that he is a very, very important researcher, scientist, and I'm very happy that you accept to give us this talk today. Thank you.
Right. Thank you so much, Dr. Rocha, for this very, very kind introduction. Every time I hear people talk about like this, that makes me very humble. And this is really about the research, not about anyone's accomplishments. But I appreciate very much having this. And let me give back the compliment. As we started talking about this and me preparing for my presentation here, looking into this, I'm very impressed how you've set up this Latin American Epilepsy Initiative here. I think these initiatives are extremely important that we as researchers, as scientists, as healthcare takers talk and discuss science, discuss how to help patients, how to improve the research. These are these international collaborations and interactions that I so really dearly love in this field, and that I think is why I ended up being a researcher, in part because of all these interactions. So with that said, let me maybe go to my first slide and explain to you how this connection between me and Dr. Rocha actually started. So this shows me here with a student, Sukriti. Sukriti was in my laboratory as a high school student for 2 years until she graduated last year. But she was still very eager to present her research, and so she decided to attend the American Epilepsy Society meeting last year in Atlanta. And so Sukriti, here in front of her poster, presented on cannabidiol treatment and sort of the role of some of this at the blood-brain barrier. And after the meeting, she reached out to me and said, Dr. Bauer, I was contacted by a researcher from Mexico who was very interested in this cannabidiol research. Would you mind getting in touch with her? And so I did. And this is how Dr. Rocha and I got in contact. And so after several months, now here I am. Thank you again for this kind invitation to present here. Very excited, very eager to present. And these are the kinds of things why researchers go to meetings, talk to each other, to start these kinds of interactions. So again, thank you very much for having me here. So let me jump now to my sort of first slide, give you a little bit of an overview on what I actually do. let me preface everything that I say that I'm a blood-brain barrier researcher. I'm not an epilepsy expert. So I look scientifically at the world through the lens of the blood-brain barrier. And what you have here in the middle is essentially a cross-section through the blood-brain barrier, through the endothelial cells, with the lumen here in the middle, endothelial cells, pericytes, and astrocytes. So my view on the scientific world goes through sort of this lens of this blood-brain barrier. And as you can see, the research that we do is on epilepsy, cannabidiol, Alzheimer's disease with epilepsy, and Alzheimer's disease in general. Some of these are my main projects. On some of them, I am a co-investigator. And so for the purpose of this talk today, I will be focusing on the role that epilepsy plays in the blood-brain barrier and how seizures make the blood-brain barrier dysfunctional. This is what I will be talking about today. So let's go and hop first into what is the blood-brain barrier. So here on this slide, what you can see here on the left, this rotating 3D video, this is where we took a mouse, euthanized the mouse, took the brain, and then sliced up the brain into thin slices. We then made the brain transparent with a method called clarity. And then in that cleared brain slice, we then stained collagen that's surrounding the endothelial cells. And I'm stopping this here, stopping this here, and you will see this vast network of capillaries in the brain of this mouse here. This is what fascinates me so about this organ, the blood-brain barrier or the neurovasculature, this very tight and dense vasculature here. So analogous to the mouse, we have something here that's similar in human. This is a cast of the human brain vasculature. This is work that was done by Berislav Slokowicz 26, 7, 8 years ago. And same thing here, you see how very dense that vasculature is. So by estimates, to give you some numbers and facts, by estimates, the adult human brain has about 100 billion brain capillaries. That is an enormous number. And interestingly, that number about matches with the estimates that are there for the number of neurons in the adult human brain. So the estimates there are somewhere in the 87 to 93 billion ballpark. But what that indicates is maybe with 100 billion capillaries and about the same number of neurons, maybe that indicates that each neuron has its own capillary that provides it with oxygen, with glucose and other nutrients, and then also takes away metabolic waste, CO2, and so forth from that neuron. So this is again only speculation, but it's a very interesting speculation to play with. The blood-brain barrier itself, again in the adult human brain, is if you combine all the capillaries together, they sum up to about a total of 600 kilometers. That is a long, long distance for that vasculature, just the capillaries in the brain. And it amounts to about 20, 30 square meters of surface area. So also a very impressive and big number. Now, what we do in my laboratory, we isolate brain capillaries from mouse, rat, and human brain. And what you see here on the right, this is an isolated rat brain capillary. You see it has a diameter of about 5 microns, and that fits about in with the description in the literature that capillaries have a diameter of about 5 to 7 microns. And you can see there's here a red blood cell that just can squeeze through that capillary. So individual red blood cells can just squeeze through these tiny little capillaries. Again, this is what I'm focused on. So if we take a schematic look at the blood-brain barrier, here again, this is now imagine a capillary, and we take a cross section through that capillary. Here in the middle, this would be the lumen. This is where the blood flows. Then we have here in pink, this is the endothelial cell. And these endothelial cells at the blood-brain barrier, they're very tight. And this tightness and this tight seal comes from tight junctions that seals up neighboring endothelial cells, preventing any meaningful paracellular transport. In addition to sort of this physical barrier, the blood-brain barrier is also a biochemical barrier, which I here illustrate with these various transporters shown here. Some of them are efflux transporters, some of them are influx transporters, as we know. And in addition to this, there's also metabolizing enzymes. I depicted them here as these grayish little round symbols. So these resemble metabolizing enzymes. And so we know that these are all present there, constituting a metabolic or biochemical barrier, if you will. Now, in white here surrounding the endothelial cell, that's the basement membrane and then the extracellular matrix that is shared with pericytes that are also surrounding the endothelial cells. And then surrounding all of that are astrocytes shown here in blue. These are the astrocytic end feet that cover the vast majority of the surface area of this structure. And then in the literature, you also see that there's most likely innervation into this complex. This could be interneurons, for example, that are in contact with all of this. And so together today, we're looking at this 4-cell structure consisting of endothelial cells, pericytes, astrocytes, and neurons. We refer to this as the neurovascular unit. And essentially think of the neurovascular unit sort of as the functional unit where barrier function sits in the brain. This is about what my research was dealing with when I was a PhD student and a postdoc. And I read papers on the blood-brain barrier and on efflux transporters until I came across an interesting set of papers that described how the blood-brain barrier, specifically efflux transporters, are changed at the blood-brain barrier in epilepsy. And so some of these papers are shown here. Some of these papers come from the Wolfgang Löscher and Heidrun Poczka laboratory, as you see here. Dr. Rocha and Lazarowski have also published on efflux transporters at the blood-brain barrier. But sort of this field goes back to a initial publication by Tischler et al. In 1995, where these researchers found that in tissue that was resected from patients with refractory epilepsy, they found an increased expression level of the gene. Back then it was called MDR1, now it is called ABCB1, and this is the gene that codes for the efflux transporter P-glycoprotein. And so I was fascinated by this and was wondering, so how comes that this transporter, which back then my work was really focused on, that this transporter's levels are changed in patients with epilepsy? Well, this was about 2003, 4, 5. And then in 2005, I was a postdoc at the NIH here in the US already. I went back to Germany and attended a conference on pharmacology. And at that conference, I met Drs. Wolfgang Löscher and Dr. Heidrun Poczka. And the conversation with these two very prominent researchers in the epilepsy field changed the trajectory of essentially my scientific career. With their input, I started looking much more into the role seizures, epilepsy plays, and what effect it has on the blood-brain barrier. And as I started reading, I learned that there are more than 50 million patients worldwide that suffer from epilepsy, and that about 2/3 of these patients are lucky because they respond to pharmacotherapy with anti-seizure drugs. However, there's about 1/3 to about 40% of epilepsy patients that unfortunately do not respond to therapy with anti-seizure drugs. These patients suffer from drug-resistant epilepsy, or also referred to as refractory epilepsy. And so In the field, there are several hypotheses and theories that try to explain the phenomenon of drug resistance in epilepsy. And so several years ago, with a student in the laboratory, Fei Tang, we wrote this review paper summarizing all these hypotheses, highlighting them. But the one hypothesis that early on caught my eye was the transporter hypothesis. And the paper by Tischler et al. That I just talked about several minutes ago is really the initiation point for this hypothesis. Now, with all the research that we have done over the last 20, 25 years, most researchers will probably agree that the transporter hypothesis, as it was originally put together, is probably not anymore, while very prominent, probably does not fully explain drug resistance, which is the same for these other hypotheses. Each of these hypotheses have some advantages and some disadvantages. But with me coming from the blood-brain barrier transporter field, this hypothesis was fascinating, and I started looking into this. And today we can probably say this is still an interesting and valid hypothesis, although I would make the case we probably need to expand this hypothesis if we want to explain certain phenomena that we observe. So I started research on epilepsy and its effect on the blood-brain barrier. And on the next slide, I will essentially summarize in very briefly what we have done over the last 20 or a little over 20 years, actually. So as a blood-brain barrier researcher, the question that really interested me was, how do seizures in epilepsy change transporter expression at the blood-brain barrier? And so in the work over those last 20 some years, we essentially found a very intricate and detailed signaling pathway. And for the sakeness of time, I'm not going to go into all the details of this signaling pathway. I'll shorten it a little bit for easier understanding. Essentially, we found that seizures change the expression of efflux transporters, influx transporters, metabolizing enzymes like cytochrome P450s. They also lead to barrier leakage. And so these changes together, barrier leakage and these biochemical changes that we observe after seizures, we summarize this in the field as barrier dysfunction. Now, what we believe is that barrier dysfunction, at least in part, contributes to anti-seizure drug resistance on the one hand, but also contributes to seizure genesis, more seizures. And if you see how this pathway here goes, from seizures to barrier dysfunction to resistance and back to seizures, you see that we have a positive feedback loop on this side, but also a positive feedback loop from seizures to barrier dysfunction to more seizures over back here, another positive feedback loop. So what we believe is that we have two feedback loops that are tied together. And if you know what positive feedback loops mean, they're essentially, except for 2-3 exceptions, they're almost always pathological. They indicate progression of a disease. For the purpose of my talk here, I will not speak on this part of this feedback loop. This is a topic that I will address in detail at this ILAE meeting in Mexico in August. What I will be talking about here today is here this left part of this pathway, how seizures lead to barrier leakage and how this barrier leakage contributes to more seizures, and targets that we could potentially use drugs for to stop barrier leakage, to repair barrier leakage, and hopefully stop seizures. So this is what I will focus on today. So let's start with what is barrier leakage and when did we find this historically? So if we go into the literature, the earliest literature references on barrier leakage in seizure or after seizures is actually from 1958 from two clinicians that were described this phenomenon that seizures lead to an increased blood-brain barrier permeability. This was described at the University of Heidelberg, which is actually my alma mater, where my academic career started. There are some descriptions later on in the literature, but up to this point, this was essentially only an observation in the clinic. And interestingly enough, researchers, basic researchers, were not paying attention and did not really seem to pick this important information up until the late 2000s, when the group around Erwin van Vliet, Eleonora Aronica, and Jan Gorter in the Netherlands published this paper in 2007 describing barrier leakage in temporal lobe epilepsy. And from that point on, other researchers addressed this as well. Alan Friedman, Uwe Heinemann, Nicola Marci, Chaitali Ghosh, and Damir Chanigro here in the US. And so around that same time, 2005, 6, 7, as I got into this field, this is when I started paying attention to barrier leakage. So what is barrier leakage? In order to demonstrate that, let's go to some data that we have from patients with epilepsy. So I'm here at the University of Kentucky, where we have access to brain tissue from patients with epilepsy, and we took that brain tissue and isolated the brain capillaries of these patients. At the same time, we also have access to their serum. And what we first did, we used patients' serum and looked at this marker called S100 beta. S100 beta is a protein that is expressed in astrocytes, as you see here on the left in the schematic. Astrocytes are shown here in blue. When the barrier becomes leaky, which is indicated here, this S100 beta that is expressed in astrocytes can actually leak into the blood, and you can then measure it in the blood. And so we did that. And so you see here in this figure, in patients with epilepsy that actually had uncontrolled seizures, we see an increase in S100 beta serum levels compared to normal control individuals. Interestingly, when we took serum samples from patients with epilepsy, but they had controlled seizures, we did not see such a big difference. In fact, it was non-significant. We did have patients where we saw some high values in S100 beta levels, and we could only speculate whether these patients might have had a breakthrough seizure here or there, and that seizure was not noticed. But essentially, on average, there was no difference in S100 beta serum levels between patients with epilepsy with controlled seizures and normal control individuals. We then started looking at the blood-brain barrier after this phenomenon that we saw in serum. And what we saw at the level of the blood-brain barrier, we saw a change in tight junction proteins. So ZO1 or Claudin 1 and Claudin 5 are all tight junction proteins that in healthy patients make sure that the barrier is very tight and not leaky. But you see in patients here with epilepsy that had seizures, and this is Western blotting, by the way, that signals of these tight junction proteins are significantly decreased, potentially suggesting that there is a leakage at the blood-brain barrier, that the tight junctions are gone and now the barrier becomes more leaky. So we then went on and tried to find out how comes that tight junctions are low at the blood-brain barrier of these patients. And we found and looked into so-called matrix metalloproteinases, specifically MMP2 and MMP9. These are the two major matrix metalloproteinases at the level of the blood-brain barrier, and these MMPs are little enzymes. They're like, for the students, if you know the computer game Pac-Man, the Pac-Man that chews up kinds of things, this is what these MMPs do. They chew up the extracellular matrix, they chew up basement membrane proteins, and they chew up tight junction proteins. And if MMPs should be elevated in patients with epilepsy, one could conceive that these enzymes could chew up the tight junction proteins and through that make the barrier leaky. So we looked into this and we saw indeed MMP2 and MMP9 are expressed in brain capillaries of patients with epilepsy. We see an increase in MMP2, MMP9 by ELISA assays in patients versus controls. And we also saw an increase in overall MMP activity in patients with epilepsy versus control individuals. So with this and other data, we then formulated a hypothesis, which I'm going to show you here on this slide. Essentially, we formulated the hypothesis. This is again based on literature and some of additional research and data that we generated, that seizures release glutamate. And this glutamate, again, signals through all kinds of steps, but for clarity, I'm only going to present you here a shortened pathway. This glutamate leads to a release of arachidonic acid, and arachidonic acid is a pro-inflammatory marker that is used by enzymes like 5-lipoxygenase and cyclooxygenase 2. I will use them to refer to them here in this context as LOX and COX. And these two enzymes transform arachidonic acid into inflammatory mediators like prostaglandins and leukotrienes, and then through further signaling pathway, they lead to changes in the biochemistry of the blood-brain barrier. In other words, an upregulation of efflux transporters, a downregulation of influx transporters, an upregulation of metabolizing enzymes, as well as lead to barrier leakage. And as I mentioned before, these changes are overall summarized up as barrier dysfunction that then can contribute to ASD resistance and seizure genesis. Again, I will not really talk about this right side of the arm. I will mostly talk about here on this left side of the arm. And so this was our hypothesis, and we started doing experiments to sort of answer this big question mark here, if this was really the pathway. And so what I will walk you through now in those next couple of slides are several models that we used to address and tackle that particular question. And so the first model we used, because this is an easy model for us to do, is to take brain capillaries mostly from rat, ex vivo, and then incubate these isolated capillaries from rat with glutamate. Glutamate essentially mimicking a seizure event, if you will. So we isolated brain capillaries and exposed them to glutamate, more specifically to 100 micromolar glutamate for 30 minutes. Then after 30 minutes, we took the glutamate away, we washed the capillaries and exposed them to buffer, Dalbeco's PBS, for an additional 5.5 hours. So the total experiment was essentially 6 hours. After those 6 hours, we then incubated capillaries in a fluorescent dye called Texas Red. And what happens in that 1 hour that we expose capillaries to Texas Red is Texas Red diffuses in and in part also transported into the lumen of the capillaries where it accumulates. And then after 1 hour, we take the Texas Red away, and then with confocal microscopy, we follow and monitor individual capillaries. And we essentially take pictures at certain time points and we watch, does the capillary lose the fluorescence in the lumen? In other words, if the capillary is very leaky, the dye will diffuse out faster versus a capillary that's not leaky. And so then we have those images, we quantitate those images, and we get figures like the one shown here in this middle. So what you see here in black, this here is the data we got from control capillaries. So capillaries that are very tight, that are not leaky, and you see we get a certain level of fluorescence there. But safe to say, these capillaries retain fluorescence in their lumen. In capillaries that are exposed to glutamate, which is shown here as red, you see the fluorescence goes down dramatically and is at a very low level. And this is a level that is about comparable to what we see when we expose the capillaries to 100 millimolar mannitol. We use this high concentration of mannitol to osmotically disrupt the blood-brain barrier and use this as a positive control for maximal barrier leakage. And you see maximal barrier leakage gives us this level down here. And so glutamate causes about what looks like maximal barrier leakage in these capillaries. So we see that glutamate directly causes barrier leakage. We also used these capillaries exposed to glutamate and looked at tight junction proteins. And again, here you can see that the tight junction proteins seem to be decreased in their expression levels. At the same time, MMP2 and MMP9 seem to be increased. Levels seem to be increased. Again, this data suggesting with a lot of other data that we did that I cannot show, possibly 20 years worth of data, you'll have to take my word for this, suggesting that this increased level of MMP chews down the tight junctions and makes the barrier leaky so that dye then can leak out. Okay, so we've done this. And then we also looked at MMP by ELISA. And you see that exposing capillaries to glutamate increases levels of MMP2, MMP9, and it also increases MMP activity. If, however, we use a pan inhibitor that sort of is a cross inhibitor for most MMPs, we can blunt that effect, and inhibiting MMPs does not increase activity. So indicating this is an MMP specific effect. And then again, using that same inhibitor, if we expose capillaries to glutamate with or without this inhibitor that inhibits MMPs, we see that glutamate, big leakage, but with the inhibitor, we're almost back to control levels. So it seems to be that MMPs are responsible for the leakage in these capillaries when they're exposed to glutamate. So with this, we then hypothesized that COX-2, cyclooxygenase 2, this very important inflammatory enzyme, is involved in this process and leads eventually downstream to an increased expression of MMPs. So we repeated some of these experiments that we did. We used capillaries, exposed them to glutamate. We always see a nice increase in MMP2 and 9. And we used celecoxib, which is a specific inhibitor for COX-2. But even though we use celecoxib to inhibit COX-2, that did not seem to affect MMP2 and 9 expression levels in Western blotting. Neither did celecoxib seem to fully repair barrier leakage that we see in our assay. You see here, celecoxib does not bring the curve back up to what we here have in the control capillaries. So we were for some time a little bit stuck on this because we thought that COX-2 will be the answer to our problem here. So that did not seem to be the case until one day we realized we did not see the forest for the trees because glutamate leads to a release of arachidonic acid, and arachidonic acid signals through both COX-2 as well as 5-LOX. And so we started to look at 5-LOX instead. Again, we repeated the experiment. We exposed capillaries to glutamate. We see a nice increase, but when we use silutin, which is an FDA-approved 5-LOX inhibitor, again, we did not really see an effect on MMP2 and MMP9 levels, and the same was true in our barrier leakage assay. Silutin did not repair barrier leakage. You see here this red line, silutin did not prevent barrier leakage. It's not here with the control levels. So at that point, we were scratching our heads and we were thinking, if it's not COX-2 and if it's not 5-LOX, what could it be? And we realized that we probably need to inhibit both 5-LOX and 5-COX. Maybe through both pathways we have an increase in MMPs. And so now we did the experiment inhibiting both 5-LOX and COX-2. And we did this with a compound called licofilone. Licofilone is a dual LOX-COX inhibitor. And using licofilone and inhibiting both LOX and COX, we now saw that we could blunt the effect that glutamate had on MMP and MMP2 and 9 levels. You see here glutamate increases them, Licof alone blocks this effect, and we don't get this increase in MMP levels. And we repeated that also looking at MMP activity, and you see here Licof alone also blunted that effect. More importantly, when we used Licof alone in our barrier leakage assay, you can see that Licof alone restored barrier integrity. It repaired barrier leakage caused by glutamate, which is shown here in red. So with this, we then knew we have to block both COX-2 and 5 LOX. Inhibiting only one of them is not sufficient to repair barrier leakage. So all of this work that I showed you so far, this was done in rat. So we went back using human brain capillaries and using the Xyluten and celecoxib. And I want to make the point here that both of these compounds are FDA-approved drugs that are on the market. Patients are taking them. Celecoxib for rheumatoid arthritis and Xyluten for asthma. And so we use these drugs to mimic what Licof alone did as one compound. And just before I go into those data, I want to show you both 5 LOX and COX-2 are expressed in human brain capillaries. And then here we did the barrier leakage assay. Instead of a time course, we did a one-time snapshot, which is shown here. And so again, Texas Red was incubated with capillaries. Some capillaries were exposed to glutamate, and you see in the capillaries exposed to glutamate, we have very low Texas Red fluorescence levels in the lumen. Xyluten itself has a little bit of an effect. Celecoxib has a little bit of an effect, but neither of them fully restores barrier integrity. Only if we use both Xyluten and celecoxib, we can get back to control levels and repair this barrier leakage. So all of this data is done from isolated capillaries. Let's switch to another model. This is now an in vivo model in rat that suffered from acute seizures. This is essentially a pilocarpine model where we use rats that were injected with pilocarpine repeatedly until they develop a status epilepticus. And then once these animals had status epilepticus, we give for 90 minutes, we give them diazepam, stop the status epilepticus, and then 2 days later, we euthanize the animals, we isolate capillaries and analyze these capillaries. And similar to what you've seen before, here you see animals that had status epilepticus, so very, very heavy seizures. You see there's a significant decrease in levels of tight junction proteins. You also see there's an increase in S100 beta serum levels in those animals. And you see that barrier leakage is quite big in these animals. So the fluorescent compound is very low in the lumen of these capillaries, indicating barrier leakage in animals that had a status epilepticus or acute seizures. In addition to looking at these, we also looked at MMPs in animals that had status epilepticus. Here you see MMP2 and MMP9 expression at the level of the blood-brain barrier of rats. You see an increase in MMP activity in animals that had acute seizures. We also see a slight increase in animals that were given pilocarpine but did not have a full status epilepticus. These animals, interestingly, they had a few seizures, but not a full status epilepticus. And so what that indicates to me, even a few seizures seem to increase MMP activity. And in a way, you could say here, you have something like that resembles like a dose response a little bit. We looked at MMPs by Western blotting. You see here this increase in MMP2 and 9 levels, animals that had status epilepticus. And then in addition to this, we did an experiment. This was done in mouse, in mice, where we gave them pilocarpine to induce status epilepticus. And after they had status epilepticus, we gave these animals this compound called MMP Sense. MMP Sense is a small peptide where you have a fluorophore, then a little peptide linker that's an MMP substrate, and a quencher. And as long as the quencher is close to the fluorescent substrate, there's no fluorescence. When MMP activity is high, MMPs can cleave that little peptide linker, and the quencher goes away, and then the compound becomes fluorescent. And so what you see is in animals that are controls, there's very low MMP activity compared to animals that had pilocarpine injections with some seizures, and then animals that had full status epilepticus. You see a full lighting up of fluorescence in the brain of these animals. This was done in vivo. These animals were alive, and this is done with in vivo imaging. So all of these data indicate that also in an acute seizure model, animals that had status epilepticus, we see the same changes that we've seen in some patient samples as well as in ex vivo samples. So then we used these acute seizure rats and were doing in vivo experiments, first using celecoxib blocking COX-2. And what you see there in animals that had seizures, I'll start here with MMP2 serum levels. We saw an increase in MMP2 and in MMP9 levels, but we did really not see much of a change when these animals were given celecoxib. Similar to what we've seen before, celecoxib itself does not really seem to have an effect. That seemed to be sort of similar when we looked at S100 beta levels in these animals. These S100 beta levels are high compared to controls. Celecoxib itself does not bring levels back to control levels. And so the same is seen with the barrier leakage. Again, this is barrier leakage just looking at one time point. Very low fluorescent levels, so barrier integrity is very low. Celecoxib does not fix this, and you see here maximum leakage here with this. So you don't really see much of that change with celecoxib. So then we repeated that experiment with Silutin, and we inhibited 5 locks. Again, looking at MMP2 serum levels, there was really not much of a decrease in MMP2 serum levels with Silutin. Same is true for MMP9 here on the bottom right. Same thing for S100 beta serum levels. Seizures increase S100 beta serum levels. This is a typo here. This should say Silutin. Silutin does not change this level. So we cannot reduce S100 beta serum levels. And the same is true for barrier leakage. Silutin does not repair barrier leakage itself. So then we did the experiment where we combined both. We inhibit COX and LOX with a combination therapy of Silutin and celecoxib. And now we see what we wanted to see. Now we see that with both inhibitors, we get the MMP2, the MMP9 levels back to control levels. We get the S100 beta levels back to control levels, and we can repair with the dual inhibition, we can repair barrier leakage. So this seems to be working in an acute seizure model. And then the last couple of slides that I have here for you are from a chronic epilepsy model. This chronic epilepsy model is a model where we induce acute seizures in animals, a status epilepticus with pilocarpine. After 90 minutes of status epilepticus, we give the animals diazepam, and then we let the animals sit for 5 to 6 months until they develop what is referred to as spontaneous recurrent seizures. And we monitor these animals, and we can then select animals that have spontaneous recurrent seizures from ones that don't. And the animals that do have those seizures, that is referred to as a chronic epilepsy model for the purposes here. And so let me show you the data that we have collected from our chronic epileptic rats. So animals that have chronic epilepsy, again, you see here their tight junction levels are reduced compared to control animals. Their MMP9 level is significantly increased compared to control animals. Their S100 beta levels are significantly increased compared to controls. In addition to this, we also looked at the barrier leakage by doing 2-photon in vivo imaging, where we create a cranial window in a rat, and then with the microscope literally look into the brain of these animals. And so what we do in these animals, we give them an intravenous injection of a fluorescent compound. And you see here in control animals, the fluorescent compound is contained to the vascular space. In animals that have chronic epilepsy and seizures, several seizures a day, the barrier seems to be leaky and the dye leaks from the blood into the brain. You see there's a very distinctly different staining and coloring pattern here. And sort of just to give you a little bit of an idea of other things that we also look at, we've looked at a whole battery of inflammatory markers. This here is just looking at one of them, interleukin-17 alpha, that we have a more recent interest in. You see a dramatic increase in interleukin-17 alpha levels. With autoradiography, we looked at TSPO that can sort of be considered general as a marker for neuroinflammation. And between control animals and animals that have chronic epilepsy and have seizures, you see everything here in green essentially represents TSPO, indicating there's not just inflammation at the level of the blood-brain barrier, but really neuroinflammation all over the brain. And one thing that we have an increased and recent interest in is also looking at adhesion molecules in the blood-brain barrier. And you see in animals that have chronic epilepsy a dramatic increase in ICAM1, so one of the many adhesion molecules there are. This is important because these adhesion molecules can be used by immune cells in the periphery to dock on and bind to them and then make things worse. They also release, immune cells also release MMPs that can create more barrier leakage. They release inflammatory marker and so make this overall inflammation worse. And so we have an increased interest now here in looking at adhesion molecules and seeing what we might be able to find there. But with all these observations, the important question is, what happens if we dose these animals with our two inhibitors that block LOX and COX? So again, we have COX-2 and 5-LOX, this time here shown in red, expression at the blood-brain barrier of these rats. We dose these animals twice a day for 2 weeks with 5 milligrams per kilogram Silutin and 10 milligrams per kilogram celecoxib, and then looked at barrier leakage. And as you can see here, dual inhibition of LOX-COX repairs barrier leakage. It restores S100 beta levels back to control levels. It also restores here, this is soluble ICAM1, which essentially is sort of a marker for neurovascular inflammation, restores those to control levels, as well as a general inflammatory marker, TNF alpha, is also restored to control levels. And in addition to this, we also looked at several other markers. This is sort of just a snapshot to give you a flavor of the observations that we have made. Then on my next slide here, you see MMP2 serum levels back to control levels with the dual inhibition. MMP9 back to control levels with the dual inhibition. And then we were interested in looking at whether or not this dual inhibition itself could sort of reduce seizure burden, where we monitored animals for 2 weeks by motion sensor and by video 24/7 for 2 weeks, and then analyzed their seizure burden expressed here per day. And you see there was a slight change just using these inflammatory markers in reducing the seizure burden, although I do not believe that this by itself would ever be a good anti-seizure medication. You would really have to combine this with anti-seizure drugs, and this is the process that we're doing right now. We can demonstrate that, which I have down here, LOX-COX inhibition repairs barrier leakage, and we are now in the process of combining this with anti-seizure drugs to see, can we get seizures in animals that have chronic epilepsy? Can we reduce this and get seizures low down to ideally 0 seizures. We will see what we will find. So to summarize what I showed you that seizures release glutamate, and this glutamate signals through two very important inflammatory pathways, 5-LOX pathway and the COX-2 pathway, leading to changes in the blood-brain barrier biochemistry, leading to barrier leakage, and that combined causes barrier dysfunction, which on the one hand, we haven't really talked about, this leads to anti-seizure drug resistance, and on the other hand, this leakage leads to more seizures, feeding back into more seizures, and so we have a positive feedback loop. I also showed you that when we target 5-LOX and COX-2 with Silutin and celecoxib respectively, we can stop what it looks like. We can stop this cycle and at least repair barrier leakage on this end. And the question remains now for us, if we use this dual treatment in combination with anti-seizure drugs, will this potentially help tackle anti-seizure drug resistance? So this is my last data slide. I want to thank members of my laboratory, in particular Manekya Sumitra Rachi, who's done quite a bit of work on this, all the helpers who are in the lab helping with all these experiments. In fact, right now, as I'm talking here, my team is in the laboratory generating status epilepticus for generating more chronic epileptic animals. I also want to thank past members, in particular Ralf Rempe, Brent Sokola, who really contributed dramatically to the lab's progress. My main collaborator, Annika Hartz, who, full disclosure, also happens to be my wife. We are a husband-wife research team. I have a number of collaborators here on campus who help with various things. Sanders Brown Center on Aging, this is the center that gives us the human brain tissue, all the funding resources, and then, of course, all of you who were listening to me over this last 45, 50 minutes or so. And I'll switch to the last slide. If anyone has any questions about how we do the things in the laboratory, the research, any of the data, or anyone is interested in maybe a research opportunity with us, please feel free to reach out to me directly. Here's my email address. If you put my name into Google, you can probably find our website and get in contact with us through this. So with that, again, thank you very much for having me. It's a great pleasure to be here. Very exciting. And I will be happy to answer any questions that you may have. Thank you very much.
Thank you so much. Now I have some questions in YouTube.
Yes, go ahead.
Liliana Quintana. Are MMP2 and MMP9 zinc dependent? If so, is there a loss of zinc homeostasis or an increase in zinc demand in epileptic animals?
The question, if I understand the question correctly, MMP2 and MMP9, are they independent?
Dependent or independent? What do you think?
Independent of each other?
Yes.
Okay. We don't really know this. We know that there's a third MMP called MMP13 that seems to be activating them. We have not really had any good luck looking at MMP13, sort of trying to see how MMP13 activates them. We don't know if MMP2 and MMP9 depend on each other. So you may know that in addition to MMPs, there are also other enzymes, and it's a very delicate balance on how they activate and deactivate each other. But we are not MMP experts at that level to really have all the tools. I should say there are a huge number of MMPs that affect, some of them affect each other, some of them don't affect each other, and there are lots of tools also for MMPs. The problem is that a lot of these tools are not specific for one, let's say MMP2 or MMP9. Like the inhibitor that we use, this GM compound, is really a pan inhibitor. It's not really specific to inhibit just one of the MMPs. And so we have only limited tools that we could use. But to answer your question, we don't know if MMP2 and MMP9 are dependent or independent of each other. We have not really looked at that, to be honest with you.
Thank you. We have a comment from Professor Esper Caballero from Brazil. Very elegant and didactic presentation. Thank you.
Thank you.
One question from Javier Ramos. Excellent talk. Thank you. The blood-brain barrier disruption is due to a primary endothelial alteration or a secondary effect due to inflammation?
That is a very good question. It's the question of the chicken and the egg, essentially. If you go into the literature, you will see that Alan Friedman several years ago wrote an interesting review article about this, where he named it, what is it? What's the chicken? What's the egg? The question about the chicken and the egg. What I anticipate is you could potentially have both as the initiation event. I could see a scenario where there's a traumatic brain injury, and there's traumatic brain injury that causes both leakage, that causes inflammation, and it makes it all worse. I could see where you have an event where you open the barrier and that causes seizure by itself. So in one of what I did not show you until several years ago, I was also involved. We had a in my laboratory also a glioblastoma group, so brain tumor group. And one of the approaches in treating glioblastoma is to inject high osmotic mannitol into the carotid to open the barrier to then deliver chemotherapeutic into the brain after surgery, after the tumor has been taken out, to eliminate and decimate all the other tumor cells that are still resident there. And these patients always get levetiracetam as a co-treatment, because when you open the barrier, that itself can cause seizures. Now you could see that something opens the barrier that could cause seizures, and then inflammation, and you have the positive feedback loop. You could see that there's an injury that causes inflammation, and that causes then that whole feedback loop. I think both could be the initiation. It's hard to say which is the primary cause on a case-by-case basis.
Okay. My personal question is about mast cells attached to the blood-brain barrier. We have found that immediately after the seizure, acute seizure, there is an important release of histamine and also many other substances that are included in the mast cells. My idea is that glutamate activates and provokes that mast cells are disrupted, and that provokes the damage of the blood-brain barrier.
I think you have a very good point there as well with the histamine. I have a colleague who has an interest in migraine, who's also been looking at histamine and mast cells and sort of that. I think that role, and I'm not very versed in migraine, looking at that. And you may know there's a 2007 paper, Fabene et al. In Nature Medicine. Now, they did not specifically look at mast cells, if I recall correctly, but they looked at sort of immune cells, sort of generally. And if they take away the immune cells one way or another, they dramatically reduce damage and also seizures. But I think you have a very good point there with the histamine. We have not really looked at this.
Okay.
The colleague, I don't know if you've ever looked at mast cells, histamine, and migraine. The colleague who is doing this, he himself has migraine. He's using now sort of our approach of inhibiting COX with an NSAID and 5 logs, he uses an I think a leukotriene receptor antagonist, montelukast, and he said he has never had a single migraine again after using this. It's an N of 1. I don't know what it means. I'm happy it helps him, but I find it interesting as an observation itself.
Thank you. From Leonor, at the molecular level, where do you think these two pathways converge to produce an effect that is not detectable when either pathway is inhibited individually?
If I understand the question correctly, the question is, how are these pathways connected?
Yes.
So let me, I did not really show this vast signaling pathway. I have it right here in front of me on my pin board here. So I'm reminded of it every day. But let me walk you through this. What we have observed at the level of the blood-brain barrier. Everything I showed and that I'm talking about is at the level of the blood-brain barrier. I cannot speak for other brain cells. We have looked at endothelial cells. We see that when seizures happen or when we expose them to glutamate, glutamate signals through the NMDA receptor. And yes, the NMDA receptor is expressed at the level of endothelial cells. When we published this 17, 18 years ago, there was a lot of skepticism. A lot of people have now shown NMDA receptor is there at the level of endothelial cells and functional. So signal through the NMDA receptor. What we believe happens, obviously, NMDA receptor opens, calcium floods in. Calcium activates cytosolic phospholipase A2. You can see what I'm talking about. Most of this is published. You can see this in the literature. The activation of cytosolic phospholipase A2 leads to a cleavage of arachidonic acid. And arachidonic acid can then go one of two ways, through the LOX pathway or through the COX pathway. The COX pathway, we see a generation of prostaglandins, and those prostaglandins, mostly then the end product prostaglandin E2, is transported by a transporter MRP4 out of the cells, where it can then, in an autocrine or paracrine fashion, bind to EP1. We've shown it binds to EP1 at the level of endothelial cells. And then from there, downstream signaling leads to NF-kappa B, and we see that that changes efflux transporters. You know, as an example, efflux transporters are upregulated by that. On the other side of the pathway, LOX, 5 LOX is activated, generates leukotriene A. Leukotriene A can go in 2 pathways. It can go and be converted by leukotriene B synthase into leukotriene B4, which itself is a chemoattractant for immune cells. And on the other side of that, leukotriene A4 is converted into cysteinoleukotrienes. That's leukotriene C, D, and E4. They bind to cysteinoleukotriene receptors that are also expressed at the level of endothelial cells. And first we thought, we didn't know about 5 LOX. We only knew about COX-2 and thought that is the answer to sort of the question in solving this. But it didn't work. None of the experiments we did worked. And when we looked into the literature, there's a good body of literature trying to inhibit COX-2 alone, and it doesn't help. Some of these animals even have more seizures. And that makes sense, because if you block COX-2, now the whole arachidonic acid shifts over to the 5 LOX pathway, and then you get barrier leakage through that. And so we did both of these experiments until we figured out we have to inhibit both. And it seems to be, that seems to do the trick to sort of stop that flow down of these pathways. I hope that answers the question. I hope that was not too specific, but that sort of gets you at the molecular level, if that was the question.
Okay, there is another question. From the perspective of the blood-brain barrier dysfunction in epilepsy, do you think that prenatal stress could alter blood-brain barrier maturation and thereby contribute to the increased seizure susceptibility observed in neonates?
Absolutely. That's a very, very good question. Prenatal stress, and what that could do. Now, with that said, um, we only have a little bit of data on this, especially in humans, you can imagine. It seems that the blood-brain barrier, prenatal blood-brain barrier, and then also shortly postnatal, so perinatal phase, um, is not fully developed yet. So we don't know exactly what needs to happen. So people are in the field are now looking at blood-brain barrier development, in that natal perinatal phase and how long it takes. Different people have different opinions on this and slightly different data, but newborns don't seem to have a fully developed barrier yet, and it takes some time for it to fully close and shut down and be very, very tight. If that is true, you could imagine that if there is an injury, let's say lack of oxygen, how that could potentially affect and cause injury that will then be longer lasting. So I absolutely believe that this could be a problem, prenatal injury that could lead downstream to issues, including then seizures.
Uh, Other question is, in the developing brain, is the interaction between these two signal pathways preserved in the mechanisms in underlying blood-brain barrier disruption associated with epileptogenesis?
Another very, very good question. I can tell these are epileptologists or epilepsy experts who ask these questions. I don't know the answer to this. Um, Let me say that the blood-brain barrier field per se, the core blood-brain barrier field, is a fairly small field, and only very few people really look at blood-brain barrier development, let alone prenatal or perinatal development. We don't. I am not aware that anyone has looked at this particular question. That sounds like somebody could have an entire career out of this and look at this. This is a really important question, in my opinion, to look at and find out, could there be prenatal injury that leads to epileptogenesis and then to a lifetime of epilepsy in kids like that or individuals like that. I don't know the answer. I don't know if anyone knows the answer.
The, does, does celecoxib reduce P glycoprotein expression?
Correct, it does. So this is so we've done. I have not shown any of those data, but yes, if you use celecoxib in these animals or in the cells in the isolated capillaries, the increase in PGP is fully blunted, goes completely down to control levels.
Given that CBD has well-documented neuroprotective effects, do you think that part of this effect could be related to the modulation of the blood-brain barrier integrity during epileptogenesis?
Absolutely. So that very first picture I showed of the student with me, Sukriti, she analyzed some of the data, and at that same meeting, I presented something on endocannabinoid signaling. What we see, this is a project that started during the pandemic when a colleague asked me, "Hey Bjorn, cannabidiol is now legalized here in the US. Would you be interested in looking at cannabidiol in your animal models?" And so we did. And not only is cannabidiol sort of related to this, cannabidiol actually is involved, has a connection to LOX and COX. Cannabidiol um, downstream downregulates COX and LOX expression levels. And so we have now already done several experiments combining LOX COX inhibition with CBD and looked into sort of endocannabinoid signaling in general. There's nothing known about that at the level of the blood-brain barrier and into the role CBD plays. But to your question, I absolutely believe that and we have looked at CBD. CBD lowers everything that we see in terms of inflammation, and it seems to repair the blood-brain barrier as well. So this is what we see in our experiments. Absolutely.
In the case of status epilepticus during early development, could the damage of the blood-brain barrier affect hormonal control in adulthood?
Could the damage of the blood-brain barrier lead to what?
In adults.
I believe so, yes. So we have I believe we have not done specific experiments to this, but from sort of experiments where we looked longer term effects, I would believe that could happen. Let me give you an example. We have done experiments where we give animals status epilepticus. These animals are about 8, 9, 10 weeks old. Okay, yes, they're adults. They're not neonates, but they're adult animals. We give them an injury with status epilepticus, and then we look months later, up to 12 months later, when we treated the animals early with or without inhibitors. The inhibitors in this case, we specifically use CPLA2 inhibitors. CPLA2 comes upstream from LOX and COX. We tested all this. There are currently no CPLA2 inhibitors that are on the market. There's some very, very little research on this in some other diseases. But so we were interested at the time at CPLA2 until we realized this is not the best. But what we found is pre-treating animals or not treating has an effect 12 months later. At least we know that from rats. We have only done this one experiment. You can imagine it's an expensive experiment to let sit animals there for 12 months. And over 12 months, they develop all kinds of other issues. But from this one experiment, we see that this effect has something long term, and whether we treat or not early has an effect long term as well. So again, it's not a full study. It's sort of a one N of one. But based on that, I would not be surprised if early treatment or early injury has long-term effects.
Thank you. And one question from Dr. Mónica Díaz Coranges.
Mm-hmm.
And she has the expertise in blood-brain barrier here in our department.
Yeah.
Nice talk. My question is, how can you explain that in the glutamate model, there is no change in Claudin 5 expression?
Can you repeat this? I just had a building alarm here go off.
How you can explain that in the glutamate model,
yeah,
no change in Claudin 5 expression?
Yes. So we've done this many times. Sometimes we see a little bit of change. Sometimes we see a little bit more. Sometimes we see a little less. We don't rely just on sort of one measurement. We usually measure also other things, as I showed you. We also, measure barrier leakage. I could probably show you some other Western blots where you see a little bit more of a change. But I know which Western blot you're referring to, where there was not much of Claudin 5, where we saw Z01 or Claudin changes, but not much of Claudin 5. I don't have a good explanation for this other. It seems to be variable depending on who makes an experiment or whatever specific conditions were there. You bring up a good point though, because of the tight junctions, and if you work in the blood-brain barrier field, I'm sure you know this. Right now, we think Claudin 5 is sort of a very key tight junction protein, and changes you see in Claudin 5 seem to be very relevant. It used to be Z01, zonal occludence protein 1, where people, 20 years ago, everyone thought that's sort of the key. I think that has shifted. where we believe it's really Claudin 5. I don't have a good explanation for that particular Western blot, other than this is experimental variation.
Thank you so much. I think Alberto Lazarowski, there is another. Let me see. He sent to you his best regards, Alberto Lazarowski from Argentina.
Thank you very much, Alberto. Very nice.
And thank you so much, and hope to see you soon in Mexico.
Thank you very much. Thank you much for all the students there, for everyone who was, I think, watching on YouTube, from what I understand. Thank you to you, Dr. Rocha, for having me here. This was really exciting. Great questions. And I still have this, I think, up here. If anyone is interested, more questions, please feel free to email me. We can set up a Zoom meeting. I would be more than happy to talk more about the research, about what tools we use, or any other questions anyone might have. Thank you very much, everyone.
Thank you.
Bye bye.